Hi, this is Jessica Lee. I'm the Branch Chief of Oncology Branch 2. Today, I will present early development of cell and gene therapy in oncology, clinical consideration. My disclosure. My presentation will cover key clinical regulatory issues frequently encountered during early clinical development of cell and gene therapy in oncology. Outline of today's presentation. First, I will provide a high-level overview of early clinical development of cell and gene therapy in oncology, followed by discussing key clinical considerations, including trial design, endpoints, dosing and dose escalation, and safety. Last, I will conclude with a brief summary. Overview. In early phase cell and gene therapy trials, several key factors are crucial, including number one, safety. The primary objective of early phase cell and gene therapy study usually focuses on demonstrating the safety of the investigational product. Number two, selection of the starting dose for first-in-human study. Due to the lack of clinical data, selection of starting dose for first-in-human trial will be based on extrapolation from animal studies. Number three, dose exploration, which usually varies according to different products. In addition to determining the maximum tolerated dose, additional dose exploration strategy includes to determine the optimal dose for safety and efficacy, and to determine the feasible dose that can be consistently manufactured and administered. Number four, activity assessment. In addition to safety and dose determination, consider to evaluate preliminary clinical efficacy and to determine if there is signal for potential benefit. Number five, manufacturing feasibility. Due to the complex and challenging manufacture process for cell and gene therapy product, consider incorporating assessment of manufacturing feasibility into study objective. In the next section, I will provide high-level coverage on study design, endpoints, dosing and dose escalation, and safety. First, study design. Most of the first-in-human cell and gene therapy trials in oncology are single-arm studies and often focus on unmet medical needs, such as patients with disease relapsed or refractory to available therapies. However, for combination therapy, it will be challenging to assess the contribution of effect in a single-arm setting. The mechanism of action for cell and gene therapy often involves specific targets. Therefore, may require companion diagnostics to identify suitable patients. Depending on targets, consider reaching out to either CDIH or CBER-OBRR to validate the in vitro diagnostic assays. In addition, companion diagnostic assays may require a protocol-specific risk evaluation. For significant risk devices, IDE are required before the study can proceed. Study endpoint. When defining study endpoints in cell and gene therapy trials, consider the following. As mentioned before, most of the first-in-human cell and gene therapy trials in oncology are single-arm study, with main purpose to demonstrate the safety and to find the optimal dose for the investigational agent. Other key endpoints include tumor response rate and duration of response. There is limitation of single-arm trial in that tied to event endpoints, including overall survival and progression-free survival, can be challenging to interpret without concurrent control. When assess the endpoint, consider the impact of concurrent treatments. Patients might receive combination regimens such as lymphodepletion chemotherapy, interleukin-2, or combination with other immunotherapy, including immune checkpoint inhibitors. These factors can confound the results and complicate the evaluation of the investigational product's true effect on safety and efficacy. Dosing and dose escalation. When selecting the starting dose for early phase and first-in-human study, please provide a robust rationale and justification for the selected starting dose based on preclinical data and any clinical data from similar products of the same class. In general, selection of starting dose for first-in-human study is usually based on toxicology data and prior human experience if similar constructs have been used in previous studies. For engineered cell therapy, the dose should be based on the number of transduced cells rather than total cells. In general, dose escalation schema for early phase cell and gene therapy trials should consider anticipated cell expansion in vivo and anticipated toxicities. In addition, for cell and gene therapy, we generally recommend a half-log increment between dose levels. Regarding dose escalation schema, both the 3+3 design and Bayesian's adaptive designs are acceptable. In general, we do not recommend intrapatient dose escalation for cell and gene therapy. Last, consider implementing staggered dosing between subjects within each cohort and between cohorts. Safety. The main purpose of early phase and first-in-human trial is to demonstrate the safety of the investigational product. Ensuring safety in clinical trials involves several critical components, including but not limited to safety monitor and management plan, dose-limiting toxicity criteria, and stopping rules. Of note, the safety monitor and management plan should include duration of safety monitor, management plan for potential toxicity, and monitor for potential adverse events of special interest, if any. Now I will elaborate further on safety monitor plan, focus on duration of monitor and long-term follow-up plan. Regarding duration of monitor for adverse events, safety monitor should extend long enough to cover the expected duration of the therapy's effect. In addition, duration of monitor will depend on preclinical studies and prior safety information of related products, if any. Regarding long-term follow-up, the follow-up duration should be tailored to individual product. For example, certain cell and gene therapies, particularly those involving integrating viral factors, may require extensive follow-up up to 15 years to fully assess long-term safety and late onset effect. Dose-limiting toxicity. Regarding DLT observation period, ensure sufficient observation period to cover both acute and subacute toxicities. Please note concurrent treatment can confound and complicate DLT assessment. In general, DLTs are context-dependent and may vary by therapy, such as cytokine release syndrome and off-target toxicities. Please provide clear definition for DLT. Use established criteria like CT-CAE or AST-CT to define DLTs to ensure consistency and accuracy in evaluation. For example, for cell and gene therapy study in oncology, DLT often include grade 3 or higher major organ toxicity, provided that these are not pre-existing or not related to the underlying malignancy. Trial stopping rules. In general, trial stopping rule will trigger temporary pause in enrollment and treatment to limit exposure to excess risk. This pause is not meant to terminate the study, but to allow for thorough safety assessment. Specify well-defined conditions such as type and number of adverse events that trigger a temporary suspension of enrollment and dosing until safety review is completed. Based on the safety assessment, protocol revision may be needed. This could include changes to eligibility criteria, dosing regimen, or the safety monitor plan to mitigate identified risk. Consider include deaths not due to progressive disease within 30 days of receiving investigational cell and gene therapy, or any deaths directly attributed to cell and gene therapy as trial stopping criteria. In summary, cell and gene therapy hold significant promise in cancer treatment, with products advancing rapidly from bench to bedside. These therapies are complex and diverse, with unique mechanism of action and inherent safety concerns. Therefore, each clinical trial must be evaluated individually, considering the specific characteristics and the risk of the therapy. Thanks for your attention. This concludes my presentation. Here is a list of useful FDA resources. This is a list of FDA contact information. The next section, I will be happy to answer any questions you might have.